So there are 5 kingdoms, right? Kingdom Monera, Kingdom Protista, Kingdom Fungi, Plant Kingdom, and Animal Kingdom. So really, for the rest of this course, we're going to be talking about these kingdoms of living things. And we're going to start with Kingdom Monera. Now, Kingdom Monera is made up of all the organisms that are one cell big. They're one cell, large, and the term we use is unicellular. That means one-celled, single-celled, and they have no nucleus. They are prokaryotic, meaning no nucleus. So all the organisms that are one cell and have no nucleus are placed in this kingdom. Now, how do these single-celled organisms multiply? How do they reproduce? All they do is undergo cell division or mitosis. We had mentioned that for single-celled organisms, they don't reproduce sexually. They don't make sperm and eggs. They just divide into 2 cells, and then those cells divide. So that's how they multiply. As far as the structure, of these cells, so we wrote that they have an outer rigid cell wall. And somebody was asking me about cell walls. Cell walls are cellulose. All right, they are, in the case of plant cells, or they're polysaccharide. So an outer cell wall is always a polysaccharide. In plant cells, it's a polysaccharide called cellulose. In bacterial cells, it's a different kind of polysaccharide. Cell membranes are phospholipid bilayers and proteins. That's a cell membrane. So if this is a bacterial cell, it's got a cell membrane and an outer cell wall made up of a polysaccharide. We've talked about, and we needed to know for the test we just took today, about not only eukaryotic cells, but prokaryotic cells. They have cytoplasm, which is mostly made out of water and protein. And then there is this ring-shaped chromosome made up of DNA. Right? So we've covered all this back in section D, is when we had talked about this before. Now, on page I2, included in Kingdom Monera are blue-green algae and bacteria. You'd say, does it say that? Yeah, it says the prokaryotic organisms, we can divide them into 2 general groups, the blue-green algae and the bacteria. So what's the difference between blue-green algae and bacteria? The only difference is that blue-green algae carry on photosynthesis. So they are plant-like in the respect that they make their own food using sunlight. So they are simple photosynthetic organisms. The word that we commonly use for simple photosynthetic organisms is algae. And the reason why they're called blue-green is because they look blue-green because they contain chlorophyll. Now, the term that we use for organisms that make their own food, which plants and these guys that are like plants do by photosynthesis, is we use the term autotroph. And the root auto means self, and troph means to feed. So literally, they're self-feeders. They make their own food. In contrast, bacteria are heterotrophs. Bacteria, let's break up the word. Hetero means other. Troph means to feed. So, putting it together, they feed on other things. They feed on others. Heterotrophs. You'd say, I didn't know hetero meant to other. I mean, like, is that like heterosexual? Yeah. Somebody who's a heterosexual is sexually attracted to the other sex. Hetero means other. So, in this case, we're talking about heterotroph, to feed on others. All right, so that's really the difference between bacteria and blue-green algae. So here, both bacteria and blue-green algae are very small, single-celled organisms. The only difference is that blue-green algae contain chlorophyll, and they make their own food by photosynthesis. Bacteria do not have chlorophyll. They cannot do photosynthesis. They cannot make their own food. They feed on other things. Is everybody clear on that? So, and we have out on microscopes, and you can look at them today, and you can look at them next time we meet. blue-green algae and bacteria. Now, let's say a little bit more about blue-green algae. So the word algae, we said, is just a term we use to refer to simple plant-like organisms that carry on photosynthesis. What is photosynthesis? We've talked about this before in lecture. If you haven't yet covered it, you will in a lot of detail, including in my class. In photosynthesis, 6 molecules of carbon dioxide from the air are combined with 6 molecules of water using light energy captured by chlorophyll. The purpose of chlorophyll is to absorb that light energy. And then using enzymes, the carbon dioxide and water are joined together to form sugars. Sugars like glucose, C6H12O6. And in the process of using carbon dioxide and water to make sugars, oxygen is given off as a byproduct. This is called photosynthesis. So plants do this, plant cells, and so do blue-green algae cells do this. They carry on photosynthesis. So we've said that blue-green algae are autotrophs. They contain chlorophyll. All right, I've just underlined or highlighted certain major points. We've already talked about how they reproduce by just dividing by mitosis. On the next page, I3, now commonly when we look at a algae, and for that matter even bacteria, when they divide and they increase in number, remember they have an outer cell wall that's made up of a sticky polysaccharide. A polysaccharide is a type of sugar. So when they divide, they commonly tend to stick together, forming long chains. Does that make sense? Because they got a sticky outer cell wall. It's still considered a single-celled organism. You might say, well, isn't it now multicellular? No, because you could shake it all apart, break it all apart into individual cells. That doesn't change how they live. If you break apart all the cells in our body, we're no longer a human. These guys, it's not like these cells function collectively together. So it's a single-celled organism. It's just that as they divide, they tend to stick together. That's all, because of that sticky outer cell wall. Here, they're sticking together that way. There's about 1,500 species of blue-green algae. I'm only going to make you learn 730 of them. Now, I'm just kidding. We're just going to look at one single example of a blue-green algae. Everything that we look at in this class, when we look at an organism, we're looking at a single example of typically thousands of species. So obviously, people who get into this, there's a lot more to study than one organism. The one that we're going to look at under the microscope basically looks like this. It's going to look like little cells that are green because they contain chlorophyll. There is no nucleus in them. When you look at the cells carefully, they have no nucleus. It's a prokaryotic cell. And they just tend to stick together as they multiply, forming these long chains. And this, technically, the one we're looking at is called Anabina. I'm not asking you to know that name. Here's all that I want you to know. It is kingdom Monera, phylum blue-green algae. That's what I want you to know. So when we stop fairly shortly today, I have already set up microscopes here. And you'll notice there's under this microscope is that blue-green algae. It's going to look just like that picture. And the thing I want you to know is it's kingdom Monera, phylum blue-green algae. In other words, on a future test, there will be microscopes out, and you'll be asked to identify what are you seeing, what are you looking at, and what kingdom is it, and what phylum is it. All right? And you'll say, gee, it must be kingdom Monera. You'd say, yeah, why? Because it's single-celled organisms with no nucleus. Right? The cells don't have a nucleus. And since they're green, it must be the blue-green algae. Because if it wasn't green, then it'd be a bacteria. Now, you might ask, well, Professor Fink, even if I look at it today, we're not even going to meet for another 3 weeks. How am I going to study this at home? Oh, I'm assuming you might ask that question. Some people wouldn't even ask, I'm not going to study. Okay, let's address that. How are you going to study at home? So, if you go to my website, you will see where it says Professor Fink's video review of Kingdom, Monera, and Protista. We'll click on it. It's got almost 8,000 hits, and I posted it less than a year ago. And I haven't had 8,000 students. I only teach this class once a semester. And all right, so there's the blue-green algae. Does everybody see it? All right. Doesn't it look just like that picture? All right, no nucleus in the cells. And you should be able to answer these questions. It's an autotroph, right? Because it's green. It has chlorophyll. All right, and then we're going to go and review bacteria as well. All right, we're going to get these what you see under the microscope looks exactly like this. You'd say, how come? Because all I did was take my digital camera and photograph it. All right, so you can study this at home. Okay, we'll come back to that. All right, so on page I-4, now, on I-4, what about bacteria? Now, bacteria are single-celled organisms, no nucleus, and they come in three, and of course they don't have chlorophyll. They are heterotrophs, not autotrophs. They feed on other things. They have three basic shapes. If they are round, that's called a coccus shape. If they are kind of like a rod, that's called a bacillus shape. If they're kind of like a corkscrew, that's called a spiral or spirillum or spirochete shape. Is the shape they come in. Now again, they have a sticky outer cell wall made up of a polysaccharide, just like the blue-green algae. So as they divide, they commonly will tend to stick together. And depending upon the pattern in which they stick together, we give them different names. If we have we have round coccus-shaped bacteria that we tend to see in pairs, that's called a diplococcus, di meaning two. If they tend to form a long chain, that's called a streptococcus. You'd say, you mean like a strep throat? Exactly. A strep throat is when you've got streptococcus bacteria eating your throat up. And if they form a cluster, like a cluster of grapes, that's called a staphylococcus. Or, for short, they'll call it a staph infection. All right, that's like a cluster of coccus bacteria. Now, we see the same thing can happen with the rod-shaped guys. We can have a diplobacillus, meaning a pair, or a streptobacillus, a chain of them. All right, so what I've just told you, the fact that they have three basic shapes, is what we wrote right here. Okay, those are the three basic shapes. Now, on page I-5, on I-5, as we've said before, most cells have no color to them, and we commonly use dyes. We dye them or stain them so that they show up easier and we can study them better. And in 1883, there was a Danish doctor named Hans Christian Graham. Incidentally, my question for you is this. Is everybody living in Denmark named Hans Christian? All right, I remember Hans Christian Andersen, but anyhow, and this guy Hans Christian Graham discovered or invented this very dark purple dye called crystoviolet, and certain bacteria absorb this very dark purple dye, and other bacteria do not. And for those of you who plan to take microbiology, is there anybody in this class who plans to take micro? All right, I know for most of you you're not, but you will learn in microbiology one of the major ways we categorize bacteria is based on those bacteria that absorb this purple dye and those that don't. And the bacteria that absorb it are said to be gram-positive bacteria. And those that do not absorb the dye, the purple dye, are said to be gram-negative. All that I'm mentioning is that we use different stains to categorize bacteria and identify them. On page I-6, we're not going to do this today, but next time we meet in 3 weeks, we will give you an opportunity to look at bacteria in your own mouth. All right? And basically, the method we'll use is on I6. We'll do this in 3 weeks. Next time we meet, you're going to scrape the inside of some of the cheek cells, just like you did when you were looking at your cheek cells before. But this time, we're going to use that Gram stain, that very dark purple dye. This is actually the cheek cell. Here's its nucleus. And you will see these little, very small, round, coccus-shaped bacteria all over your cheek cells, because your mouth is loaded with bacteria, including coccus bacteria that absorb that purple dye. And you'll see just how many bacteria are in your mouth. The human mouth probably has as many bacteria as any place, probably more bacteria than any other place in your body. You'd say, why is that? Think about all the things you put in your mouth. All right, so you put almost anything in your mouth. So, and as a little kid, kids will pick up anything and stick it in their mouth. Is that right? Yeah. That's what they like to do. You know, they pick up a block and stick it in their mouth. Anything. So anyhow, we'll learn how to look at bacteria in our mouth next time. On I7, we sometimes culture bacteria. You'd say, what's that? We grow bacteria. And commonly what they'll do to culture or grow bacteria is they'll take a Petri dish with a agar, which is basically like a jello in it, with nutrients that the bacteria can feed on. So what we're looking at here is a Petri dish, and if you can see these lines on here, all they did was they took a stick and dipped it in something containing certain bacteria, and they just rubbed it back and forth, just like they did here, and then, because they were introducing some bacteria right on this jelly-like stuff, it started to grow. So this is called culturing or growing bacteria. And in fact, a number of years ago, the way they used to determine whether you had strep throat or not is they would do a bacterial culture. Remember, the doctor still even today says, open up your mouth. They put that stick down your throat, and they actually take a swab, and they swab it. And in the old days, what they actually did was they would do exactly this, and they would see what bacteria grew. And they would tell you 2 or 3 days later whether you had strep throat or not. Today, they have a biochemical test they use, which tells them instantaneously whether you have strep throat. And the reason why they want to know whether you have strep throat is they need to know what type of antibiotic to give you, and the type of antibiotic they use depends upon which type of bacteria you have in your body. So if you have a urinary tract infection, which is a different kind of bacteria, they use a different antibiotic than if you have a strep throat. So we have to know what it is you're trying to kill. All right, so that's called a bacterial culture. Now, the importance of bacteria. There are 20,000 species of bacteria. We're only going to learn 18,721 of them. Okay, we're just going to learn a few of them. Obviously, people who take microbiology or bacteriology obviously spend a lot more time learning a lot more. If there are 20,000 species, they're going to learn hundreds of them in a bacteriology course, because some of these cause bacterial infections. Now, in fact, the majority of bacteria, and let me just, here's what, you can write what I wrote right here at the bottom of the page. Okay, now, you'd say, what did you write? Bacteria are heterotrophs. You'd say, what does that mean? That means hetero means other, troph means feed. They feed on other things. So bacteria feed on other things. Most bacteria feed on non-living things. Some bacteria feed on living things. So the majority of bacteria are not feeding on you or any other living things. The majority feed on non-living things. You'd say, what do you mean? What are they feeding on? Sewage, feces, dead organisms. They feed on, let's say, dead organisms, and they cause them to decompose. These are actually considered good bacteria. We need these bacteria in our world. Because if we didn't have these bacteria that break down, that feed on dead things, that feed on feces, that feed on sewage, then the world would be piled high and deep in the remains of dead things and sewage and manure and feces. So what causes all of that stuff to break down are these bacteria that feed on the organic molecules, the organic compounds that are in the dead organisms, that are in the feces and in the sewage. So in fact, they use these bacteria at the sewage treatment plant. When you flush the toilet, that whatever you flush down the toilet is carried through a sewer to the sewage treatment plant. The closest one is Hyperion in El Segundo. And they actually dump bacteria right into this sewage, and they have these big holding chambers. They're like ponds. Has anyone ever seen this thing? They're like concrete ponds, and they add bacteria to break it down. And it's the bacteria that are feeding on all that sewage and feces and manure, and they're eating it. And basically, they do that several times. And they run through what's called a primary, secondary, and tertiary treatment. And after they're done, basically, there's no more sewage left in the water, and they actually release it right out into the ocean. It's actually suitable for drinking, but people don't want to drink it. But it's suitable for drinking. So these are considered really good guys. Now, the term that we give to these good bacteria is they're referred to as decomposers or saprotrophs. And that's what we wrote here. Saprotrophs are decomposers. Those are bacteria, we wrote, that along with fungi play a very beneficial role in the decomposition of dead matter and the recycling of minerals within the ecosystem. So everything I just told you, even about sewage treatment plants, is written there. Now, returning back to the bottom, we did say that some bacteria feed on living things. They're called parasites because they're feeding on you or your dog or your cat. They need food and you're it. And so in this case, they actually make you sick. Let's show you an example. On page I8, actually, let's look on I9. Let's look on I9 for a moment. All right? Now, here's a guy on the back of his neck. He's got some pimples. All right? Acne. What is that? So actually, if we looked in the skin, this is like a cartoon view of the skin, there's actually a whole bunch of bacteria in the skin feeding on the skin. And that's called an infection. It's all infected. These bacteria are feeding. If we squeeze a little bit of this pus and stuff from around the pimple and look at it under the microscope, it looks like this. And you'd say, yeah, what's that? Does it look like a little cluster of coccus-shaped bacteria? They call that a staphylococcus or a staph infection. That's what's causing the pimples and boils. So these are really, really small, right? So, and you will see that we put a staphylococcus under the microscope, one of them that we've got under there. And it's on that video. Going back to I8, so we wrote that a small number of bacteria absorb nutrients from larger living organisms, including people. And so these bacteria that are feeding on you are said to be pathogenic parasites because they cause you to get sick because they're feeding on you. And if you don't kill the bacteria that are feeding on you, it's either you win by destroying the bacteria or they win and they kill you because you're their food. So they create what's called an infection. So pimples and boils are caused by a staphylococcus bacteria. Staphylo means like a cluster, and coccus means little round bacteria. A strep throat is when the long chain of coccus bacteria are feeding on your throat. Now, I mentioned a whole bunch of others. And I'm not asking you to memorize all these. It's easy to remember Staphylococcus and Streptococcus. You can see, though, tetanus, tuberculosis, urinary tract infections are usually caused by bacteria. And I also like to draw your attention to gonorrhea, syphilis, and chlamydia. Those are the big three of the sexually transmitted diseases. Now, you'd say, but didn't you mention genital herpes earlier today? That's right, genital herpes is a virus. And we said, unfortunately, you can't kill a virus because they're not alive, so there is no cure for a virus. So if you got genital herpes, you don't die from it, but there's no cure. Gonorrhea, syphilis, and chlamydia are caused by bacteria. And the good news is this. They are small living cells. What's alive can be killed. And the drugs we use to kill living things are called antibiotics against life. And as we said earlier, the type of antibiotic we use depends upon which type of bacteria you have. So the antibiotic we use, for example, against, say, for example, strep throat is amoxicillin, which is a type of penicillin. The type of antibiotic we use against, let's say, syphilis might be an erythromycin, could be a penicillin V, different types of antibiotics. We use totally different antibiotics against tuberculosis. So you got to know which bacteria you're dealing with to know which chemical to use to kill it. The older name for gonorrhea, syphilis, and chlamydia was venereal diseases, or VD. If you've ever heard that term, they don't generally use that term anymore. They use the term venereal because it came from the Roman goddess of love, Venus. In other words, you contracted it by lovemaking. Today, we don't talk about making love, we talk about having sex. So they're called sexually transmitted diseases, or STDs. Just a little bit further here, and then I'm going to stop. We've covered I9. On I9, at the bottom, bacteria, we've learned, there are good bacteria that are decomposers, or saprotrophs that break down sewage and stuff. There are bacteria that cause diseases, bacteria infections, but bacteria are used commercially by us for all kinds of purposes. Bacteria are used to make rubber. Bacteria are used to make alcohol and vinegar. Bacteria are used in pickling of cabbage into sauerkraut, or pickling cucumbers into pickles, or pickling olives. All of these involve doing what's called a pickling reaction or fermentation reaction using bacteria. There are cultured dairy products. If you had me for lecture, I showed you the label of something called strawberry kefir, and it was labeled a cultured dairy product. And I asked, so does that mean it's refined and cultured and it went to finishing school? And I responded, no, that's not what they mean by a cultured dairy product. It means that it's got bacteria culture. There are bacteria in that dairy product. Now, before I tell you more about that, there are naturally bacteria in milk. There are naturally bacteria in the milk. When they milk a cow, there are bacteria in it. And it's okay to drink it if it's got bacteria, but those bacteria multiply. So, and that makes milk spoil very quickly. So almost all the milk sold in the United States is pasteurized. And it's called pasteurization. It was named after Louis Pasteur, a very famous French microbiologist, who found that if you heat the milk at 145 degrees, you don't have to know the temperature. For about 30 minutes, you don't have to know how long. But by heating the milk for a certain period of time, you can kill most of the bacteria, and that way, the milk has a longer shelf life. So when you buy milk in the market, if you buy it today, it probably says that it's supposed to be sold like in a week. And even in a week, it just doesn't mean that it's gone bad. In fact, it should last for even another week after you buy it, after the date posted on it. But that's only because the milk's been pasteurized, where they killed most of the bacteria naturally in the milk. They do sell milk called certified, raw milk, certified raw milk. It has not been pasteurized. Some people say they prefer it because they like the taste of it better. It hasn't been warmed, but it spoils very quickly. It's only good for about 3 days and it goes bad. On page I-10, let's go back to this idea of cultured dairy products. The bacteria that they usually use as a culture to make certain dairy products is a bacteria called Lactobacillus. Now, from the name bacillus, we know it's a rod-shaped bacteria. That's what a rod-shaped bacteria is called, a bacillus shape. And lacto means it's in the milk. It naturally lives in the milk. What this bacteria does, and it's considered a good bacteria, is it breaks apart the sugar in milk, which is called lactose, and it breaks the lactose down first into glucose and then into pyruvic acid, also known as pyruvate sugar. Now, if you learn all this stuff in lecture, if you have me for a lecture, we will get to all of this. I'm just slow. But everything that we're covering here, we're going to go into more detail in lecture. So the glucose is turned into pyruvate sugar, and then that bacteria, that lactobacillus, turns it into lactic acid. And this last step of turning pyruvate sugar into lactic acid is called fermentation. Fermentation is a process that occurs when there's no oxygen. Now, converting the lactose sugar in milk, which doesn't taste that sweet, incidentally, but turning it into lactic acid tastes sour. So our first thought is, why would we want to take the sugar in milk and make it turn into something, a chemical called lactic acid that tastes sour? Because that's how we create sour cream and cottage cheese and buttermilk and yogurt. And in fact, if you just have plain yogurt that hasn't been sweetened or with any fruit, doesn't it taste kind of slightly sour? All right, same with buttermilk, same with sour cream. So a lot of people like that kind of somewhat sour, tart flavor. And then you may add strawberries or whatever to it in addition. These are called cultured dairy products. They all contain this lactobacillus bacteria to convert lactose into lactic acid. Now, some people really like this stuff because they suffer from being lactose intolerant. And what lactose intolerance means is that some people cannot digest lactose that's in milk. They could as a child, but as they grow older, it's a genetic defect. It's not a serious one, but they lose the ability as they grow older to break down lactose in milk. So if they drink milk or anything with lactose in it, it causes them to get diarrhea and cramps. So they usually just avoid drinking milk or eating dairy products. But they usually can drink or eat yogurt and kefir and buttermilk and sweet acidophilus and sour cream and cottage cheese. Why? Because there's no lactose in it. The bacteria has turned it into lactic acid, so that's not a problem if the lactose has already been broken apart into lactic acid. Okay, and as I wrap this up here, we wrote that the bacteria are also used not only to make buttermilk and yogurt, they're used to make cottage cheese. Now, remember the story, little Miss Muffet sat on her tuffet eating her curds and whey. And what happens when you make cottage cheese is it curdles into these little clumps called curds. And you may have seen cottage cheese sold as small curd or large curd, cottage cheese, the size of the clumps or curds. And it forms a liquid called whey. And they actually use whey in many food products. You'll see it contains this milk product derivative called whey. But she was eating cottage cheese. If you were wondering, what was little Miss Muffet eating? She was eating her curds and whey. She was eating cottage cheese. Now, in this country, we call it cottage cheese. If it's made in France, we call it Neufchâtel cheese. And if it's made in Italy, it's called ricotta cheese. And it just has a slightly different taste because they're using a slightly different variety of that bacteria, the lactobacillus. But they're all fundamentally cottage cheese. We also use bacteria to make Monterey Jack, Gouda, cheddar cheese, Swiss cheese, and Parmesan cheese. Now, just before I summarize the Kingdom Monera, I want to return back to a page, a topic that we skipped over. Let's return back to page I-7. And back on page I-7, we pointed out that of the 20,000 species of bacteria, In fact, the majority of them are regarded as being beneficial, not bad guys, but actually good guys. We've already learned now that some of these bacteria are important in the environment as decomposers or saprotrophs, which are mentioned right here. We also learned that bacteria, they're useful bacteria that are used in the production of cheeses, in the production of sauerkraut, in the production of pickles, and all kinds of other things. But I also wanted to mention another beneficial role of bacteria, and that's that some bacteria carry on a process called nitrogen fixation. And I wanted to explain what nitrogen-fixing bacteria are. Let's take a look at a picture on page I-8. On page I-8 at the top, so what we're looking at on the top of page I-8 is a plant. And plants require nitrogen in order for them to survive. I think anybody who knows anything about plant food for plants knows that they need nitrogen in the soil. And they actually attach nitrogen atoms onto sugars to form amino acids. This occurs in plants, and then once the plants have converted sugars into amino acids, they can join these amino acids together to form the proteins that are contained within these plants. Now, interestingly, air is 80% nitrogen gas. It's only 20% oxygen. It's almost 80%, it's really 79.8% nitrogen gas. So in fact, every time you take a breath, most of what you're inhaling is actually nitrogen gas, not oxygen. And we as humans and other animals, they don't use nitrogen, they inhale it, and then they just exhale it right back out again. Only oxygen is used for the process called cellular respiration. Interestingly, though, plants are not able to use the nitrogen in the form of N2, nitrogen gas, from the air. They can only use nitrogen that's basically found within the soil. But interestingly, there are nitrogen-fixing bacteria that exist and these in the soil, and these nitrogen-fixing bacteria convert some of the N2 or nitrogen in the air into NH3, which is ammonia, as well as nitrates. And because of these nitrogen-fixing bacteria in the soil, they increase the amount of ammonia and nitrates in the soil naturally. Now, obviously, you can add ammonia and nitrate fertilizers, but these nitrogen-fixing bacteria are the natural ways that nitrogen in the air is converted into a source of nitrogen that the plants can utilize. Now, interestingly, there is a group, a family of plants that are known as the legumes. And the legume family, which includes beans and peas and alfalfa and clover, they actually have a very interesting symbiotic relationship. Now, the word symbiosis, which we're going to be talking more about in the next section, but I'll just write the word symbiosis. The word symbiosis is made up of two Greek roots. Sym means together, and bio means life or living. So literally, living together. And this is where, this is a relationship where two different kinds of organisms live together. This may be a beneficial relationship, or it may be, quote, an adversarial relationship. But in this particular example that we're going to talk about, it's a mutually beneficial relationship. And that's the relationship between nitrogen-fixing bacteria living within the roots of these bean family members, such as beans and peas and alfalfa. In this photograph, it shows the roots of a clover plant, which is a member of this family. And these bulges or bumps in the roots, these nodules, are actually filled with nitrogen-fixing bacteria. So this is a symbiotic relationship. What the bacteria get by living inside these nodules of the roots is a place to live. And what the plant benefits from this is that these bacteria are actively converting nitrogen in the air into ammonia nitrates, which provide greater sources of nitrogen to these plants. In fact, that really explains why the bean family members have the highest amount of proteins in them. So anybody who's a vegetarian knows that the foods that they principally rely upon as a source of protein, since they're not eating meat and fish, is beans, because beans are very high in protein, and that's largely due to the fact of these nitrogen-fixing bacteria living symbiotically together within the roots of these plants. Now that I've reviewed this, let's summarize Kingdom Monera in this fashion. All right, what we've learned about Kingdom Monera is that the characteristics of this kingdom is they are all unicellular, which means single-celled, and prokaryotic, which means they lack a nucleus. These are extremely very small cells that lack a nucleus and most other internal organelles. We divide Kingdom Monera into two major phyla, or subgroupings. We will call them phylum blue-green algae and phylum bacteria. That's not really their scientific names, but those are the terms that we'll give. Now, what's the difference between the blue-green algae and the bacteria? Very, very straightforward. Blue-green algae carry on photosynthesis. They are indeed blue-green in color because they do contain chlorophyll, and they manufacture their own foods using carbon dioxide and water and minerals from the soil or the water, whatever environment they're living in, along with sunlight to make their own foods. Phylum bacteria are heterotrophs. So they are, unlike the blue-green algae, which are known as autotrophs, they make their own food. Bacteria feed on other things. Troph means to feed, hetero means other. The majority of these bacteria are considered good guys. They feed on non-living organic matter, and they are referred to as decomposers or saprotrophs. There are, however, a small number of bacteria that give all bacteria a bad name because these bacteria feed on living things, and they are called parasites. And they can be responsible for a number of infectious diseases in us humans as well as other living things. That's a summary of Kingdom Monera. Just one last point. You'll notice that you might ask, why have they included plant-like organisms and, quote, animal-like organisms in the same kingdom? And the answer is because what blue-green algae and bacteria both have in common is they are both single-celled and lack a nucleus. So that's why they're grouped together.